Hello, good evening to all. At first, I welcome our honorable speaker and all participants. I would like to thank our respected principal madam, head of the Department of Chemistry, dear colleagues, research scholars, and our beloved students. I request Dr. Papia Ganguly, head of the PG Department of Chemistry, to introduce our respected invited speaker who will enlighten us over his talk on the topic spectroscopy and microscopy at the Nano-Bio interface. Please.
Thank you, Monisha. A very good evening to our honorable speaker and our participants. It is my privilege to introduce our illustrious speaker, Professor Anindya Datta of IIT Bombay. Professor Anindya Datta's educational qualifications are highly commendable. He has done his BSc in Chemistry Honours in 1992 from erstwhile Presidency College, now Presidency University, that was under the University of Calcutta. He has completed his MSc from Calcutta University in 1994, after which he proceeded to do his PhD from the Indian Association for the Cultivation of Science. The degree, of course, was awarded from Jadavpur University. As regards his academic experience, it started from 1997 when he joined Raidiki College as lecturer. He briefly served at Raidiki College till 1998, after which he proceeded to the United States of America to complete his postdoctoral research work at the Iowa State University from 1998 to 2000. He was visiting scientist from 2000 to 2002 at Raja Ramanna Center for Advanced Technology. He was assistant professor at IIT Bombay from 2002 to 7 and associate professor from 2007 to 12. And he has been there in IIT Bombay since then. He was the head from 2018 to 21. He was the chairman, Hostel Coordinating Unit from 2009 to 13, the Associate Dean of Student Affairs from 2013 to 15. He was the head Center for Research on Nanotechnology and Center for Sophisticated Analytical Instrumental Facilities from September 2016 to May 2018. He has, Professor Datta has very many awards to his recognition. He received the IIT Bombay Young Investigator Award in 2005, the Young Researcher Award awarded by Chemical Research Society of India in 2006, the G.V. Bakor Memorial Award from the Indian Chemical Society in 2011, the Departmental Award for Excellence in Teaching at IIT Bombay in 2017. He was a Fellow of National Academy of Sciences, India, FNASC, in 2017, and he received the Bronze Medal from the Chemical Research Society of India in 2018. Professor Anindya Datta has a number of national and international publications to his credit. His papers are 133 and he has 2 book chapters. He has guided 15 students to do their PhD, and 8 students are still working for PhD under him. Today, he will enlighten the audience on his topic on spectroscopy and microscopy at the Nano-Bio interface. May I now request our illustrious speaker, Professor Anindya Datta, to please address the audience. Over to you, Anindya.
This is my group from about 1 year ago, and what you see here in the photograph is us standing by power relay. So this is also sort of an interface. On this side, you have this lush green jungle, that is IIT Bombay. On the other side, you have this not so lush green concrete jungle. So that is where the rich and famous of our area stay. As I was saying, our goal is to get light out of molecules and material when we want them. So we work with fluorogenic molecules, molecules that are not fluorescent by themselves, but can be made to become fluorescent by specific physical or chemical inputs. In the middle, you see this chelating agent, which by itself is not fluorescent. However, when it forms a complex with certain metal ions, then it becomes strongly fluorescent. We'll talk about this briefly. On the left hand side, you see photographs of two flasks. One that is not fluorescent. By the way, this photograph is taken in a UV chamber. So you don't see anything in the first one. And the first flask contains a homogeneous solution of a fluorogenic molecule. It is not fluorescent in solution. However, if we add a bad solvent like water, then we get aggregates comprising these nanorods that you see at the bottom. So these are images that we have got from using a technique called fluorescence lifetime imaging and microscopy. I'll briefly talk about it, but we don't need to understand the detail. For now, it is enough if you understand that we can get images of these nanostructures under our microscope. And these structures are responsible for this kind of strong fluorescence that we see if we bring about aggregation. I'll not talk about this in very great detail because in August 2020, in the same platform, this is what we have presented in detail. So we can get fluorescence out of these molecules by complexation. We can get fluorescence out of these by aggregation. And what you see here again, ignore this data for the moment. This thing that you see here is a fluorescent powder, solid. So this is an extreme case of aggregation where the solvent has been removed completely. So by making solids, conventional fluorophores usually lose their fluorescence. Our fluorogenic molecules become more strongly fluorescent in the solid phase, and that has plenty of applications. The other kind of system, and we're going to show you some data on these today, are photoluminescent semiconductor nanocrystals, which have been touted as the, well, quote-unquote fluorophores of today for the last 20-25 years. And the intention is eventually to try and understand biological problems, biological systems, using these molecular as well as nanocrystalline probes. To do this, we need equipment, and these are our workhorses. The measurement that we do most of the time is for whatever reason I cannot get a laser pointer, but I hope you can see the cursor. So what we do is this. We try to record fluorescence spectrum, which is a plot of fluorescence intensity against wavelength or energy. From there, we determine the fluorescence quantum yield, which is a measure of how strongly fluorescent something is. We do another kind of experiment in which we excite our system with a pulse of light. Pulse means light that is usually off, goes on for a short amount of time, short span of time, and then goes off once again. So all of us have seen examples of pulses. If you have seen flash photography, the flash that is used here is some kind of a pulse. All of us, I'm sure, have seen lightning. Lightning is an example of pulsed light. The difference between the flash of a camera or the flash of lightning and the flashes that we use, the pulses that we use, is that our pulses are very, very short-lived. We typically work with light that remains on for hundreds of femtoseconds to hundreds of picoseconds. Femtosecond means 10 to the power minus 15 second. Picosecond means 10 to the power minus 12 second. And whenever we talk about picoseconds and femtoseconds, sometimes we get this question, what can happen in such a short time scale? Actually, a lot can happen. Photosynthesis, for example, is all about processes that take place in these ultra-fast time scales. And as you know, photosynthesis is the origin of all life around us. well, most of the life around us. So what we do is we excite using this pulsed light, and that creates an excited state population. This pulsed light would excite a number of fluorophore molecules, number of nanocrystals, and would prepare their excited states. And when I say a number, I mean a large number, Avogadro number, for example, a fraction of that. Then when the light goes off, all these molecules or nanocrystals that are there in their excited state are left by themselves to come down to the ground state. The situation is very much similar to putting an iron rod in fire and then taking it out and keeping it out in the open. What you see there is that the moment you take it out, at the moment that you take it out of fire, the rod would be glowing red or glowing white if you heat it sufficiently. As we keep it, the glow dies out. If you could measure the intensity of the glow as a function of time, you would see that this intensity would decay most probably in an exponential fashion. And as students of chemistry, and when I say students of chemistry, I mean students of biology also, because everybody has studied chemistry in the 11th, 12th level. So as students of chemistry, we sort of understand why the decay has to be exponential. Think of radioactive nuclei. These nuclei are also left to decay by themselves. And what kind of decay law do they follow? First order decay, exponential decay. So in the simplest case scenario, the fluorescence intensity that we produce as a result of pulsed excitation also decays in an exponential manner. The only difference in formalism between what we study in chemical kinetics and what I'm writing here is that you might recognize that this is just first order decay law. The only difference is that in chemistry, we usually write e to the power minus kT, where k is a rate constant. In spectroscopy, which is also part of chemistry, it is conventional to not write the rate constant, but rather write the time constant, which is simply the reciprocal of the rate constant. This time constant is typically called the lifetime, and there's a reason for it. It is the average time spent by a molecule in its excited state. So this is something that characterizes the decay. A shorter lifetime naturally would mean that the excited state population and therefore fluorescence intensity decays very rapidly after the light goes off. A longer population, a longer lifetime would mean that this population and therefore the fluorescence intensity would decay slowly. And that is something that determines how strongly fluorescent a molecule is. The relationship is pretty simple. The quantum yield, fluorescence quantum yield, which is essentially number of photons emitted per photon absorbed, is proportional to the lifetime. And the proportionality constant is called the radiative rate constant. Similarly, 1 minus phi f, well, phi f, the quantum yield, is a fraction. So the best case scenario is 1. So 1 minus phi f is equal to kNr multiplied by tau, where kNr is the non-radiative decay constant. When you have an excited state population, the population can decay by radiative pathway in which light is given out, or by non-radiative pathway in which the excess energy is dissipated but no light comes out. And we see a lot of that happening all around us as well. See, my shirt is colored, right? Why is it colored? Because it absorbs visible light. But it doesn't glow, does it? It doesn't glow because even though it absorbs light, the excited molecules, when they come down, they actually dissipate the energy in a non-radiative fashion. That is why it does not glow. However, I'm sure everybody is familiar with materials that are fluorescent, even if you don't know the chemical composition. If you see the construction workers, for example, wear these bright orange striped jackets, right? If light falls on those jackets, they would glow. They are made of some fluorescent material or the other, right? That's all that is there. So what we're saying here is that we work with fluorescent material, or fluorogenic material. We want to know how strongly they fluoresce. So there is an experiment by which we can determine quantum yield. The other experiment, which is a little more sophisticated, is by exciting with pulsed light and generating the lifetime. And lifetime is important because it is an intrinsic quantity and it gives us a measure of how strongly fluorescent something is. More often than not, lifetime is a more useful quantity than quantum yield, even though it's much more difficult and much more expensive to determine. To determine lifetime in nanosecond, hundreds of picosecond to nanosecond time scale, we use a technique called time correlated single photon counting, which is abbreviated as TCSPC. TCSPC has become very common and is used widely by researchers of many different varieties, not just chemists or biologists or physicists, but in many different areas, TCSPC has become a workhorse. For our purpose, hundreds of picosecond to nanosecond time constant may not be enough. Many times, we need to go down to hundreds of femtoseconds. To do that, the instrument that we use is called femtosecond optical gating spectrometer. And this is what it looks like. This here is a laser that gives out femtosecond pulses, 100 femtosecond pulses. And this here is the spectrometer in which the measurement is done. I'll spare you the detail of the experiment. Whoever is interested, I have some NPTEL lectures and offered a course on NPTEL. The lectures are freely available on YouTube. All you have to do is you have to search Anindya Datta ultra fast and you'll see I think 64 or 68 lectures. Some of them are about the equipment that pictures of which you see them. Whoever is interested in learning a little more about the equipment, you're welcome to have a look. The other kind of experiment that we do is transient absorption. See, as I said, not everything is fluorescent, but everything does have electronic ground state and excited state. That's why my shirt is colored but not fluorescent. So for these materials that are not fluorescent, or even for materials that are fluorescent, sometimes it is better to look at the color of the substance before and after excitation. And the technique by which you see how the color changes as a function of time with femtosecond time resolution is called transient absorption. More difficult, much more expensive experiment. This is the setup. This black box, literally black box that you see here, is an amplified laser. And this here is the spectrometer in which the experiment is done. You don't need to understand the detail. Today, I want to keep it, today I want to focus only on the findings and not so much on the data. Over the last 2.5 years, 3 years or so, we have been able to establish this instrument in which we can look at these lifetimes under a microscope. So these are these small little lasers that we use, and this is the microscope which we use to study very, very small volumes. So in these ultra small volumes, we can use, first of all, we can use fluorescence lifetime to get better images and to get a better understanding of the heterogeneous nature of small volumes. We can also do another kind of experiment called fluorescence correlation spectroscopy, which gives us an idea about how viscous a medium is. We'll discuss an example a little superficially about how we get an idea of how the chains of a protein move, how fast or how slowly they move under different conditions. So all these experiments can be done combining the power of microscopy and fluorescence lifetime in this instrument. And using all this, what we study essentially is excited state phenomena. And since we have a heterogeneous audience, let us just go through this slide and understand what's going on. So this is a modified Jablonski diagram. Well, Jablonski is the name of one of the pioneers of fluorescence spectroscopy. So this kind of an energy diagram is named after him. I think he drew them first. So what you see here is electronic ground state. Y-axis is energy. There is no real x-axis. At a higher energy, you have what is called the locally excited state. Locally excited state means the state that you access by direct electronic excitation. Electronic excitation means excitation by ultraviolet and visible light. And then after excitation, more often than not, the molecule undergoes phenomena or reaction or processes that it does not undergo in the ground state. For example, let us take the example of photoisomerization. Once again, since everybody has studied chemistry in 11th, 12th at least, I think we all know about cis and trans isomers. Whenever we have a double bond, we can have a cis alkene and a trans alkene. And we know that we cannot really start with cis and get to trans because it's a double bond, right? CC double bond. So you cannot really go from cis to trans so easily. However, when you do an excitation, a pi star excitation, then what happens is there is post-excitation, there is one electron in pi molecular orbital, one electron in pi star molecular orbital. So what is the pi bond order? 0. Which means the overall bond order now in the excited state is 1 and not 2. So what was double bond in ground state is a single bond in excited state. And now as we know, for a CC single bond, you can have rotation. So you start with trans alkene, perform pi star excitation, and then during its lifetime of the excited state, this trans has some finite chance of rotating, and then when it comes back to the ground state, it returns to another ground state which is cis and not trans. This phenomenon is called photoisomerization, and photoisomerization plays a crucial role in very, very important life processes like fission and something called proton pumping in bacteria. So without going into the detail of what process we are talking about, let us say some photoprocess is there. So naturally, if there's a photoprocess, it has to take the molecule from this locally excited state to another state, which I like to call nascent state, NS, which has to be necessarily at a lower energy than the locally excited state. Otherwise, why will the process happen? So now see, let us consider this situation where the locally excited state is fluorescent, the nascent state is also fluorescent. It's not very difficult to see that the energy gap associated with fluorescence of locally excited state is larger than the energy gap associated with the fluorescence of nascent state. So if both are fluorescent, then the nascent state would fluoresce at a smaller energy, that is longer lifetime, longer wavelength. So if lambda 1 and lambda 2 are characteristic wavelengths of emission of the locally excited state and nascent state respectively, then lambda 2 would be longer than lambda 1. Longer wavelength, remember, means smaller energy gap. Then the signature of the excited state process is seen in fluorescence spectra. This spectrum in green is characteristic spectrum of a molecule that does not undergo an excited state process. And when the same molecule undergoes an excited state process, it exhibits, in addition to the original emission band, a red shifted emission. We can think that this 375 nanometer band, which is there in the green spectrum, is this 375 nanometer is lambda 1, and the 475 nanometer band, which is present only when the excited state phenomenon takes place, that 475 nanometer is lambda 2. So this is some real data from many, many years ago from our lab. The other thing that you can see is, if you look at time resolved data, remember, we excite by pulse light and see how fluorescence intensity changes as a function of time in a matter of femtoseconds to picoseconds to nanosecond. If we monitor this decay at the emission of the locally excited state, lambda 1, then we see a fast decay. Remember, decay of fluorescence intensity is a measure of decay of excited state population. Now, this locally excited state has two pathways to decay. One is by its own radiative and non-radiative decays that was there, and this additional depopulation because of the excited state process that is happening here. So we see a fast decay if we monitor emission at lambda 1, which is characteristic of the locally excited state. If you monitor the emission of the nascent state, lambda 2, however, then at the moment of excitation, there is no nascent state population. The population of the nascent state increases as a function of time for some time, and then of course it has to decrease. Once again, in chemical kinetics, we have studied the time evolution of population of intermediates. Remember those reactions in which we have the starting material A, from there we go to intermediate I, and from there we go to the product P. How does the population of the initial starting material vary with time? It's a decrease, very much like what you see in the blue curve. How does the population of the intermediate change? Remember, there's no intermediate to start with. Over some time, the population of the intermediate grows from the at the cost of the population of the initial starting material. So the population builds up and then eventually decays. So if you remember that and look at this decay here, I hope it's not very difficult to understand that this nascent state sort of behaves as the intermediate. So what is the starting material here? Locally excited state. What is the intermediate? Nascent state. What is the final product when all the excited states have decayed? Actually ground state. So this is a second signature of excited state process, a fast decay in the blue end of the spectrum. When I say blue, I mean higher energy or shorter wavelength, and a rise at a longer wavelength or smaller energy. Combining the spectra and the transients of fluorescence, one can actually generate time-resolved emission spectrum. I mean, I can generate the fluorescence spectrum at the instant of excitation. Then 10 picosecond after excitation, 100 picosecond after excitation, 10 nanosecond after excitation. What you see in the last panel is an example of that. Here, the x-axis is in centimeter inverse. So, on the right-hand side is higher energy, left-hand side is lower energy. So, at time 0, you see the spectrum is at a higher energy. 3.5 nanosecond after excitation, this higher energy band has depleted and has made way for a new band at lower energy. This is a signature of excited state process. Let me talk about the first excited state process from our work. This work was started by my student Avinash, who's now a postdoc in Canada. It was carried on by Sharmishta, who's now a postdoc in our lab. Very soon we'll move on elsewhere. And Prajit, who's right now working on his PhD. So we worked on this molecule here, which has this peculiar property that if you excite it, then there is something called intramolecular charge transfer. You can think that electron cloud flows from this NH2 group towards this ring. And when that happens, there is charge separation. So in the excited state, this NH2 part becomes electron deficient, the ring becomes electron rich. You can understand that a dipole is created. This NH2 part becomes delta plus charge, this part becomes delta minus charge. So a dipole moment is created as a result of excitation. This phenomenon is called intramolecular charge transfer. So we had studied the photophysics of this molecule, and since you create a dipole moment as a result of excitation, this is a good probe for studying how much time it takes for polar solvent molecules like water or alcohol or acetonitrile to sort of reorient around this newly created dipole moment. That field of study is called solvation dynamics. So what happens essentially is that you create this dipole. At the moment of creation of the dipole, the solvent molecules are oriented in some fashion, but this newly created solute dipole now governs reorientation of the solvent dipoles so that there is an overall favorable dipole-dipole interaction. And this favorable dipole-dipole interaction lowers the energy of the excited state. As a result, the fluorescence spectrum moves towards longer wavelength, that is lower energy, as a function of time. How much time? femtosecond to nanosecond. This is what happens for this molecule here. Once again, remember the x-axis is in centimeter inverse. So energy. Right hand side is higher energy, left hand side is lower energy, longer wavelength. This is the emission spectrum at the instant of excitation. And then as time passes, after 20 picosecond, the solvent here is, I think, an alcohol. In 20 picosecond or so, you see there is a significant red shift. Red shift means the spectrum moves towards lower energy or longer wavelength. And this red shift is used to work out something called a solvent response function. C of t is a solvent response function, and it decays. It is fitted to exponential functions that give us what are called solvation times. What we need to understand now is that if these plots of C of t fall off quickly, that means solvent response is fast, otherwise slow. So what we did using this is that we tried to understand the dynamics of preferential solvation. We dissolved this molecule in a nonpolar solvent like hexane, and we added very, very small amounts, less than 1% of a polar solvent like an alcohol or acetonitride. So to start with, this is the situation. This circular sphere here is your solute. Well, I should take the name of the molecule at least, 5-aminoquinoline. And since it is in a nonpolar solvent, it is surrounded by nonpolar solvent molecules. And you have these polar molecules represented as these dumbbells. Dumbbells mean, well, dumbbells with two different colors essentially represent dipoles. So one side is plus, one side is minus. So this is the situation to start with. You have very small amount of nonpolar solvent molecules. So the solute molecule here, which is not polar to start with, is by and large surrounded by nonpolar solvent molecules. Upon excitation, charge separation takes place. So the solute now becomes dipolar. What happens afterwards is that in response to creation of this dipole, solute dipole, all these polar solvent molecules which are present in very, very small amount try to rush towards this newly created solvent dipole and try to solvate it in order to achieve favorable solute-solvent dipole-dipole interaction. And once again, that leads to a shift over time of the fluorescence spectrum towards lower energy. This phenomenon here is called preferential solvation. Preferential solvation means even if the solvent molecule is polar solvent molecule is present in small amount, it is going to preferentially come and locate themselves around the solvent, the solute dipole moment, solute dipole, so that energy stabilization can take place. Well, stabilization can take place. So what we have done is that we have generated this time resolved fluorescence spectra. We have performed an in-depth study of the dynamics, and we have figured out, as is shown in this cover picture published last year, that it takes about 1 nanosecond for this entire process of preferential solvation to take place. Moreover, it is not just electrostatic interaction, not just physics, and that is where we get interested. See, interaction between a plus charge and minus charge is physics. But things like hydrogen bonding or a chemical reaction, something like that, this is chemistry, and this is what really gets us excited. So what we were able to show is that it is not just electrostatics. Specific chemical interactions like hydrogen bonding have a major role to play in the time that is required for this polar solvent shell to form around this newly created dipole in a predominantly nonpolar bulk. So that is something that we were able to work out, and we are very happy about this paper, not only because it was considered to be sufficiently interesting to be featured on the cover of Journal of Physical and Chemistry B, but also because it was selected as part of this virtual special issue of Journal of Physical Chemistry, that is being published to honor Professor Kankan Bhattacharya, with whom I had the good fortune of doing PhD. Both these cover pages of our paper and the script, special issue, are designed, by the way, by Sharmishta. She is very good at these things, in addition to being an excellent student. So this is one thing that we have done. We have figured out, generally, if you take an alcohol or water, solvation takes something like maybe 1 picosecond, 2 picosecond, maximum 10 picosecond. What we have shown is that if you take small amount of polar solvent, large amount of non-polar solvent, the process of solvation is slowed down by orders of magnitude. And such slowing down for a long time, and well, we talked about Professor Kankan Bhattacharya, a lot of work from his lab has been towards this, that when you take a biological system, like a protein or membrane, water associated with these biomolecular systems actually exhibit very slow dynamics. Hundreds of picoseconds, almost to nanosecond. So, and lots of work has happened in this area over the last 25, 30 years. Now, what we have tried to do recently is to study a rather unique biomaterial in collaboration with our colleague, Professor Kamendra Sharma. Anushwa was Kamendra's PhD student. She graduated recently. And Tarashankar was my postdoc. So they are the ones who started the work. Now, Kamendra here is expert in this very strange kind of system where one takes a protein and tries to wrap it in a polymer. And then the polymer that is used to wrap the protein is actually liquid. The next thing that they do is that they remove water from the system completely. The result is a highly viscous liquid. So you have the protein in a liquid form, thanks to it being wrapped in the polymer, which is a liquid, but there is hardly any water. So these are called waterless liquid proteins. And many times when we try to talk about these, people who are into protein chemistry do not like it, because they feel protein is such a beautiful molecule, proteins are such beautiful molecules. Why do you want to insult them and mess them up by adding this polymer surfactant and removing water? Well, the thing is this: in this kind of materials, actually the structure of the protein is retained, and in fact even function is retained. It has been shown that if you take an enzyme and make a waterless liquid protein out of it, all this work has been done mainly in the lab of Professor Stephen Mann in Bristol. It has been shown that the entire structure is retained, and moreover, since water is not there, you can try to enhance their catalytic activity to higher temperatures. So our interest was that we'll take these liquid proteins and we'll try to see solvation dynamics. So the signature of biological water, slow solvation dynamics, do we see it here or not? And the question is important because there has been a lot of debate about what is this slowness due to? Is it because the water molecules are hydrogen bonded to the protein surface? Is it due to the motion of the proteins or what? So our first attempt at this, published in 2018, was we tried to use well-known solvation probe, coumarin 153. We bound it non-covalently to bovine serum albumin and prepared this liquid protein. What you see here is a photograph of the liquid protein. You can see it's very, very viscous and a little colored. So here we did not really see any signature of solvation, presumably because this is something that is attached non-covalently, this coumarin molecule. So the moment you take out water, it migrates to the hydrophobic core, hydrophobic pockets inside, and it does not experience any polar environment at all. We did make some interesting statements about this system in this paper, but then our original intention of studying solvation dynamics was not fulfilled. More recently, my student Tanuja, in collaboration with Anushwa and under the guidance of Kamendra, has done this. She has taken a human serum albumin that is covalently labeled by a coumarin molecule. by using something called CPM, a Coumarin derivative. And what she has been able to see is this. First of all, if you do fluorescence correlation spectroscopy, well, now if you look at the time scale, we are not talking about picosecond, nanosecond, femtosecond anymore. We are talking about millisecond, and 1000 millisecond is actually a second. Now we're talking about slow dynamics. So from this data, we can work out how much time it takes for the chains of the protein to move, how much time it takes for the entire protein to diffuse. And what you see is that, so these are data taken at different stages. First, you have the native protein. Then you denature, you cationize it, make the surface little positively charged. Then you add this negatively charged surfactant, polymer surfactant. Finally, you remove water. So what you see is that the dynamics changes a little bit throughout, but it becomes much slower, significantly slower the moment you remove water, which is not surprising because after all, the viscosity increases. But then there is more than what meets the eye here. From a careful analysis of this data, we could actually make a statement about this interchain motion as well. What kind of motion changes under which condition? And finally, we could see that there is slow solvation dynamics here. And this slowness actually persists over nanoseconds. So unfortunately, this figure is not labeled and I am colorblind, so I cannot tell you which is which. So I hope you can follow the cursor. So this decay is for the native protein. The moment we cationize it, there is a fast component that comes in because it's a little denatured. When we add the polymer surfactant, you see the decay becomes a little slower. Why? Because now you have this protein covered by surfactant, so it is not very easy for it to move anymore. The point to note is that in the wrapping process itself, most of the water on the surface of the protein is replaced by the polymer chain. And finally, when we remove water, it becomes very, very slow because it's a crowded environment. So the point we are trying to make here is this. The slow solvation, which is conventionally ascribed to biological water, is observed in waterless conditions as well. Why do we observe this slow solvation? Because here, since it is covalently bound, it is covalently bound to the surface, the fluorescent probe. So first of all, it experiences this actual biological water. So there is slow dynamics. Then biological water is replaced by the polymer. Now the polymer itself has its negatively charged groups, which forms sort of a solvent shell around this coumarin moiety. And these themselves have their fluctuations, which get reflected in the slow dynamics of the protein. So the point we are trying to make here is that the slow dynamics can be observed even in absence of water, and one can correlate it to phenomena like chain dynamics of the protein. All right. I think I will skip the discussion of fluorogenic molecules completely because otherwise it will become too long. I don't want to speak for maybe more than 10 minutes from now. Otherwise, we'll lose the audience. Let us talk about the semiconductor nanocrystals. Once again, from the physics or chemistry that we have studied in class 11 and 12, we know that when we talk about solids, we don't talk about molecular orbitals anymore. We talk about bands. And I'm sure all of us have studied this correlation between bands and molecular orbitals. When you have two orbitals, from which we generate molecular orbitals by linear combination, we get two MOs, bonding and anti-bonding. If we take 4 atomic orbitals and generate molecular orbitals by linear combination, we get 4 molecular orbitals. So if we take 1 million atomic orbitals, we are going to get 1 million molecular orbitals. And I'm sure we have all studied how the spacing gradually decreases, and finally in solids you get bands. And I'm sure all of us know that in conductors, the Conduction band and valence band is the band that contains the electrons. Conduction band is the band that is empty. So sort of the band that is made up of antibonding orbitals, we can think. So in case of conductors, there is no band gap. The conduction band and valence band overlap with each other. That's why they are good conductors. In insulators, there is a large band gap between the valence band and the conduction band. That's why they're insulators. It's not so easy to promote an electron from here to there. For semiconductors, the band gap is small. That's why they can become conducting by applying a little bit of bias or by using light. So now, if you start with a semiconductor like cadmium selenide, cadmium sulfide, many such things, and start breaking it down, then what happens? Essentially, the number of orbitals participating in forming the band decreases. So there will come a limit below which we are going to see discrete energy levels in conduction in the valence band as well as the conduction band. Right? So this is what happens when quantum confinement takes place, and that is why semiconductor nanocrystals are so very interesting. So they are say 10, they are crystals of say 10 nanometer, 20 nanometer size. You can see them under electron microscope. However, they behave sort of like molecules because there are discrete levels. You might be wondering why I have drawn the valence band and conduction band as parabola. It is parabola because there is something called a momentum vector. Right? So the energy is proportional to k square. K is the momentum vector. So as you understand, k square means that is the equation of a parabola. But right now, it's not so important. What is important is this. What happens when we shine light on a semiconductor nanocrystal? As you understand, an electron is promoted from the conduction valence band to the conduction band. And when it is promoted, it leaves behind a hole, deficiency of an electron in the valence band. And this electron and hole are correlated. Together, they form what is called an exciton, unit of excitation. Then many things can happen that can break the correlation between the electron and the hole. See, when we talk about crystals, an ideal crystal would sort of go from minus infinity to plus infinity. But actually crystals terminate somewhere, right? And there are defects. So these defects form what are called trap states. There are states that can trap the electron. Even before that, if we perform an above band gap excitation, that means the excitation energy is more than the band gap, then you end up creating what is called a hot electron. Hot electron means an electron that has more energy than what you require to promote it to the band edge. So the hot electron relaxes to what is called the band edge, the lowest energy state in the conduction band, in about 0.3 to 2 picoseconds. It's an ultra fast process. And then it can actually be trapped by these defects that are there in the semiconductor nanocrystals. This electron trapping typically takes place in 2 to 30 picosecond time scale. The hole can also be trapped, but it is a faster process. And then you can have deep trapping as well. Deep trap means traps that have energy that is substantially lesser than the energy of the conduction band. So all these phenomena can take place. So we were very late entrants in the field. We decided that we are going to work with exotic nanocrystals. And we started this work in collaboration with Professor Rohit Srivastava of Bioscience and Bioengineering with the express intention of devising biological applications. But as of now, we are very far away from it. So Shucheta was my PhD student who's now an assistant professor in SP Postgraduate College in Pune. And Bala was another PhD student who's now graduated and is a postdoc in the US. So Shucheta, with help from Bala, learned how to make these nanocrystals. And we could prepare two kinds of nanocrystals by playing around with the conditions. And we learned how to make them strongly photoluminescent or weakly photoluminescent. The problem was that even the best nanotetrapods that we prepared had really weak fluorescence, weak photoluminescence quantum mean. 0.06 is nothing to write home about. So we decided to try a new strategy. It was started by Sucheta, completed by Faryad, another PhD student who will hopefully graduate this year, with help from Sharmishta. What they did is that they learned how to dope these cadmium selenide nanotetrapods with copper. And please note, this is copper 1, Cu plus, not Cu2 plus. Upon successful doping by copper 1, we could get this redshifted, relatively intense band. Quantum yield here is 30% compared to something like 1.7% or 6% best case scenario that you observed in undoped tetrapods. And to cut a long story short, using our time resolved spectroscopic methods, we could figure out what exactly happens after excitation and why is it that the copper doped nanotetrapods are so much more photoluminescent compared to undoped ones. We worked out the time constants associated with trapping. We worked out the time constants associated with tetrapods. So we were successful in building a complete picture of the photoprocesses that take place. And this gives us an idea of what kind of strategy one can use to enhance the photoluminescence of nanocrystals like this. Now, since we want to get to the bio-nano interface, obviously we need tetrapods that are water soluble. The tetrapods that I talked about so far are all soluble in organic solvents. That is because, see, all nanocrystals are covered with some molecule or the other. It can be a polymer, it can be a protein, it can be some small organic molecule, or some things. So the capping agent that we had used originally was mercaptopropanic acid, MPA. Well, sorry, the original capping agent was oleic acid, which does not really want to mix with water. So we did something called ligand exchange, by which we could replace oleic acid by mercaptopropanoic acid, which is much more hydrophilic, and we were successful in preparing quantum dots, nanotetrapods that were soluble in water. It broke our hearts to see that the moment we make them water soluble, they are no longer photoluminescent. This work was also carried out by Sucheta with help from Tanuja, another PhD student, the one who is our protein expert. So we showed that these tetrapods can denature proteins, so anything that is bound to the protein can be set free, but that is really not such a great story. So in our quest for water soluble nanocrystals that was fluorescent, Shomudipto, a postdoc who is now doing a second postdoc in Spain, and Sharmishta once again, figured out a way. Well, the synthesis, most of the synthesis is reported in literature, except for this doped tetrapod. So they figured out a way of making cadmium sulfide quantum dots that are prepared in water, and these not tetrapods, sorry, quantum dots. So these quantum dots are actually very strongly photoluminescent. You can see this photograph here, how brightly they are glowing, and the lifetime that you get is in tens of nanoseconds. Remember, longer lifetime means stronger fluorescence also. And once again, using all our femtosecond spectroscopic methods, we could work out the dynamics of all photoprocesses that take place here. And the one proof of concept application that we have seen is that the photoluminescence of these nanoparticles is quenched selectively by lead. No other metal ion that we study, which is sort of good because as we know, lead is a toxic material. So sensing lead is something that people are worried about. But why is that so? One thing I forgot to say is that they use a different capping agent. The capping agent they use is polyethylene imine, a polymer. And it turned out that it is the polymer that gives the specificity of this cadmium sulfide nanoparticle towards lead. Now, PI can actually bind most of the metal ions. So the metal ions don't reach the surface of the quantum dot. In order to quench the photoluminescence, they must reach the surface. Lead does not like to interact with PEI. So it actually finds its way to the surface, and we figured out that there is an ultra-fast hole transfer followed by an electron transfer that causes the photoluminescence quenching. So specificity is because of chemistry, downstate chemistry, that PEI does not like to bind lead, and the excited state process that is taking place here is not just electron transfer, it is preceded by a very efficient hole transfer. All right. Other work is going on using other CDS nanocrystals. Hemen Gogoi is a student who is doing it. Shanmishra has been able to prepare copper-doped cadmium sulfide nanocrystals in water. So they are even more strongly fluorescent, strongly photoluminescent. And what you see here is we are trying to use fluorescence correlation spectroscopy to understand the dynamics of photoprocesses that take place here. So this is a story in the making. To conclude, we try to engineer radiative and non-radiative processes, and we try to get light, or we try to quench light emission as and when we require. Using it, the processes that we have studied are slow solvation in solvent mixture, which is an interesting phenomenon because this is not really something that has the surface of protein or any such thing. And also we see similar slow solvation in so-called waterless liquid proteins. I have not talked about Schiff bases today, I have not talked about DBNPT. In semiconductor nanocrystals, we have established the fact that using complementary spectroscopic techniques, one can actually generate the picture, the entire picture of what exactly is going on. Let me stop here by thanking all students, present and past. Many of them have become independent scientists and doing very well for themselves. Some are postdocs, some are still continuing. Let me thank all the collaborators, postdocs, and funding agencies. Thank you very much for your patience, and I hope that we'll meet like good old days, perhaps in your campus or in this beautiful campus of ours. Thank you very much.
Thank you, sir, for your lucid talk, and it has enlightened us. The session is now open for interaction. Students, feel free to approach sir for any silly questions you have or anything you want to know, because if you want to pursue your further studies or your PhD work in this field, is really happening field, you can question sir. You please raise your hand one by one, or you can ask.
Please feel free to ask any question that might come to your mind.
Exactly.
Two kinds of audiences don't ask questions, those who have understood everything and those who have understood nothing. So we'll assume that everybody has understood everything, and now I can ask questions and conduct a quiz. Shall we conduct a quiz now?
Students,
they can also put their questions in the chat box. I think
sure.
I think they would feel more comfortable with that.
They can do that.
Whichever way they want to interact.
Yes.
And the other audience also. I mean, we have.
I think we can assume everybody has understood.
No, sir, there is a question.
Why is photoluminescence so important in this time-resolved spectroscopy? See, very good question, Srita. The thing is this. As I said, not everything is photoluminescent. And when we look at photoluminescence, we look at it against a dark background. The problem with seeing color is that in order to see color, you must have a lot of light. However, first of all, photoluminescence usually takes place at a longer wavelength. So you can excite by ultraviolet light, which our eyes don't see, and you can look at the visible light that comes out. So that's easier because you are measuring against sort of a dark background. However, if you want to measure absorbance, you want to measure it against a bright background. So photoluminescence is actually a more sensitive technique. That is why it is so popular. When you do time resolved spectroscopy, you can do two things. You can look out for the light that comes out and how its intensity falls off with time, or maybe grows initially and then falls off with time. Or you can look at how the color changes. Both are important. So what we have tried to show is that the good thing about looking at color and not photoluminescence is that, as I said, everything has some color or the other. We might be able to see the color, we may not be able to see the color, but it is there. So transient absorption spectroscopy, which looks at the color change and its dynamics, is a more general method. Everything shows up there. That is the strength and that is the difficulty also, because the signal is very, very complicated. Photoluminescence, however, is a more picky technique. Not everything is photoluminescent. So if you can do both back to back and then compare your results, then and then only you can generate a complete picture. So that has been a major theme of our work. Very nice question, Srijita. I hope you're answered.
From Pitha Samanto, I can see. Yes.
Why the electron get trapped at the energy state which has almost the same energy. So it depends on the, so that is the chemistry, that is chemistry. So there are what are called dangling bonds, discontinuities in the crystal, which can be of a chemical nature that is a little electron deficient. So they would like to attract electrons. Of course, if it's at higher energy, then electrons won't go there. Or there can be defects that are electron rich. They would like to trap holes. So depending on the chemical nature of the defect, there would be electron traps or hole traps. I hope you are answered. If not, please do ask later. So Anjali raised her hand and then she left the meeting also. Anyway, is there any other question?
I think Anjali got disconnected. She
Yes.
often does.
Yeah. I think she wanted to ask something because she had raised her. Anyway, while we wait for her to come back, in case there's any other question, I'll be happy to answer. Doesn't look like.
I don't think, students, there is another question from Sunova.
Yes.
Sir, I think you can see the question.
Yeah. Could you tell us about the fields and industries where nanoparticles find their maximum importance? So that, now, nanoparticles, for the last 30 years or so, nanoparticles have been touted as a solution to everything. And for good reason. Because see, the thing is, they have many, many unique properties. We focus only on the optical property. We are interested in nanoparticles because, first of all, they are color tunable, and the photoluminescence is also color tunable by simply changing the size. Sunova, I don't know if you are a BSc student or an MSc student, and if you are familiar with this particle in a box model. Using that very simple quantum chemical model, quantum mechanical model, one can, by and large, explain the color of nanoparticles. And even in the CBSE textbook, that figure is there, I think, that as size changes, the color also changes. So that's a very convenient thing. So that is one thing. Secondly,
she's a BSc student and they have done particle in a box.
All right. So in that case, you would understand completely. So here the particle is the exciton and the box length is the diameter of the nanoparticle. So that is what we are interested in. And then you can try and play around with them. With molecules, molecules are flaky. You take a fluorophore, try to do some chemical modification, more often than not, they cease to be fluorophores or their property changes completely. However, in nanoparticle, by doping or by changing the size, you have more handles to play around with. So that is our interest. But there are many other applications. You might have heard of super hydrophobic surfaces. So if you have nanoparticles on a surface, the surface area increases, right? And it is possible by playing around with the chemical nature once again to make them this surface so hydrophobic that no water would be there. So that is another kind of application. And then nanoparticles, end of the day, are very finely divided particles. So they can act as catalysts, and that is another very important application. I have mentioned only 3. There are many more. So an entirely different world opens up when you go down to this nanometer length scale. That is why they are so interesting, and they are finding applications in industry also. You might have heard about things like quantum dot television, right? It's no longer just in the textbook. Quantum dots are actually being used to make displays for television, right? So many such applications are there. Konkona has asked, how time resolution can we obtain with TCSPC? I encourage you to please invest maybe 1.5 hours and go through the lectures that I mentioned. Go to YouTube, search Anindya Datta Ultrafast, look up TCSPC, because that is something that will, if you want to understand exactly how it is done experimentally, that requires at least half an hour. So please do see those lectures. If you have questions, please feel free to write to me. And well, I'll type my email ID in the chat box. I'll be happy to answer more detailed, more specific questions over email. All right. Next, we have Srijita. According to the discussion today, we observed that we dealt with femtoseconds. So can we go beyond femtoseconds? Oh, excellent. Excellent question. Lovely question. We can. And we means not us, not in our lab, but we, the human beings, have. About 20 years ago, the attosecond barrier was broken. Attosecond means 10 to the power minus 18 second. Even in India, some attosecond labs have started coming up now. The reason why there has been much greater activity in nanosecond, picosecond, and femtosecond time regime in chemistry especially is that, see, and this is something that I discussed in the last lecture that we had in Bombay College. The time that is required to break a chemical bond is of the order of 100 femtosecond. And for us chemists, the most fundamental process is breaking of a bond. Yeah. So once we achieve that, we are sort of happy. But then there are phenomena like state-to-state dynamics that take place at even faster time scale. So the state of the art right now is attosecond. Very difficult, extremely costly, cumbersome experiment, but they can be done. They are being done in different parts of the world, including India.
Hello, Professor Anindya.
Yes.
I am Mustafizur Rahman, faculty of this department, Lady Bombay College. Thank you for nice talking. I have a silly question. Do we use any nanoparticle in our daily life?
Yes, we do, sometimes knowingly, sometimes unknowingly. So in Ayurveda, for example, you know that all these Swarna Bhashma and all that are used. So all of them are actually nanoparticles. And applications as day to day as shirts that are going to be spill free. They are now marketed, right?
Yeah.
For example, I'm very bad at that. My colleagues make fun of me When anybody spills tea on their shirt, they say that this fellow has done an Anindya. So the thing is, now people have invented fabric that whose surface of which is covered with hydrophobic nanoparticles. You spill tea, no problem, just brush it away and it will fall off. There are umbrellas that are available, not very costly too, which have this super hydrophobic surface. You go out in the rain, come back, close the umbrella, shake once, it is completely done. So these are some examples that are there, and then I already talked about quantum dot television.
Okay, thank you. Thank you very much.
I have a question.
Sukannadi.
That is, you spoke about this proteins having a waterless environment and they retain their structure, even enzymatic activities.
Yes.
So the question is that, is it true for all proteins or certain specific ones?
Certain specific ones, but the number is not very small. In fact, there have been studies by Kamendra and Somir Maji of our Bioscience Bioengineering department, and also somebody else from that or someone, where they have shown that if you take intrinsically disordered proteins that don't have secondary structure by themselves and wrap it in polymer surfactant and then remove water, order is induced in these IDPs as well. And the reason is brute force. What you're doing is you are sort of forcing the protein to get cramped. And when it does that, it is forced to sort of form helices, and these helices are forced to sort of come close together. That is what gives rise to secondary and tertiary structure. And when it is a globular protein to start with, it already has a preferred secondary and tertiary structure which is well known. So even after removing water, because of this, we can say environmental pressure, they end up retaining most of the structure. They don't retain all the structure. Some structure is invariably lost, but by and large, yes, they do.
No, but sometimes for, in order to exhibit their enzymatic activity, water may be essential.
In such cases, of course, there will be no enzymatic activity.
Of course, yes. That is, okay, thank you.
Sritama has asked a question, does nanoparticle photoluminescence any application in the development of quantum computing? Do quantum dots count in this? Yes, they do. There are attempts to use quantum dots in quantum computing as well, but it is not necessary to use quantum dots. She is asking how much important nanoparticles are in the field of drugs and medicine. As I said, nanoparticles are seen as elixirs. So right now, it is a question that we don't know the answer to, but potentially they are important. One thing is that these are very finely divided particles. So you can use them as drug delivery reagents. That is something that, so in fields of molecular medicine, they have immense potential. It may not be that you want to take nanoparticle as medicine as such, but using nanoparticles to deliver drugs in specific parts of the cell. That is where that is where a lot of work is going on. Of course, the problem with that is that nanoparticles also have this propensity of messing things up, denaturing proteins and so on and so forth. So that's a very active area of research.
There is another question by Shreya Ghosh, our MSc student.
Where is it?
She's kept in the chat box.
Oh yes, I missed it. Sorry about that, Shreya. It may not be quite related, but can this idea be used in protein modification, like reconstruction? Which idea, Shreya? Can you unmute and elaborate, please? Because we missed your question at that time. So which idea are you talking about for protein modification? Are you talking about this making protein polymer surfactant composites?
Yeah, protein molecules are normally very large, but if you use in a nano form, then might be the route of administration might be easier. So if this kind of idea we can use, like reconstruct a whole protein in a plasmid like that.
It is, attempts are being made, but one problem that is there is that more often than not, proteins get denatured when you add nanoparticles. And that is often a shady area of many publications. People talk about protein nanoparticle interaction all the time. The initial confusion I always had is that what is bound to what? Because see, people say that I have a 5 nanometer, a nanoparticle of 5 nanometer radius, and it binds to human serum albumin. Now, what is the size of human serum albumin? What is the size of protein? How will a protein bind to human serum albumin? Perhaps the opposite thing can happen. In fact, there are studies in which protein corona, formation of protein corona nanoparticles is a very active area of research. We are also doing something towards that. So the answer to your question is yes, possibly yes, there is significant activity in this direction.
Thank you, sir. But you just said about forming a protein corona. So are we using absorption here or something else? And how this protein corona is being formed?
You mean adsorption, right? Yes. So it's like this. See, nanoparticles are, well, at the risk of sounding repetitive, nanoparticles are very small particles, right? They have a large surface area. So when the surface area is large, then the propensity of adsorption is naturally more. Secondly, one can try and use capping agents that would like to interact with certain parts of the protein, more specific interaction. So both these strategies are used, sometimes to differing extents, to form protein corona.
Any more questions, students? Any more questions? Or you can write directly to sir to the email ID provided here in this chat box. Please take a note of it. And Shukun Nadi?
Okay, so I think we have come to the concluding part of today's program. On behalf of the organizing committee, I would first like to thank our Madam Principal, Professor Shudi Sarkar. She always encourages such academic programs, and it is definitely, we are definitely motivated by such encouragement, which she is very generous about always. I would like to thank Professor Anindya Datta. It has been an excellent talk, and he has explained things in such simple language. I'm sure all our students have been extremely benefited by his lecture. I would also like to thank our head of the department, Dr. Papia Ganguly, for her involvement and cooperation for organizing this program. Definitely the Department of Biotechnology for their funding, because of which we can make such, stage such programs. I would also like to thank Mr. Arvind Prasad for providing us with the technical support for the smooth running of today's program, and the other members of the organizing committee for today's program. The student participants, the audience, you have actively participated in asking important questions, and I'm sure you have learned a lot also through the interactive session also. So a big thank you to all of you for your participation, active participation. And I think we are almost at the end of today's program. So over to Monishadi to conclude it.
Thank you, Sukarna. This ends our program. Thank you all. Thank you, sir, for your illuminating talk. I am sure all of us have been enriched with the knowledge of the webinar. Thank you.
Thank you very much, everybody.